The endomembrane system and proteins. The endomembrane system, endo equals within, is a group of membranes and organelles. Figure 1. In eukaryotic cells that works together to modify, package, and transport lipids and proteins. It includes the nuclear envelope, lysosomes, and vesicles, which we've already mentioned, and the endoplasmic reticulum and Golgi apparatus, which we will cover shortly. Although not technically within the cell, the plasma membrane is included in the endomembrane system because, as you will see, it interacts with the other endomembranous organelles. The endomembrane system does not include the membranes of either mitochondria or chloroplasts. Art connection. If a peripheral membrane protein were synthesized in the lumen, inside, of the would it end up on the inside or outside of the plasma membrane? The endoplasmic reticulum. The endoplasmic reticulum, single quote, figure 1, is a series of interconnected membranous sacs and tubules that collectively modifies proteins and synthesizes lipids. However, these two functions are performed in separate areas of the the rough and the smooth respectively. The hollow portion of the tubules is called the lumen or cisternal space. The membrane of the which is a phospholipid bilayer embedded with proteins, is continuous with the nuclear envelope. Rough The rough endoplasmic reticulum, RER, is so named because the ribosomes attached to its cytoplasmic surface give it a studded appearance when viewed through an electron microscope. Figure 2. Ribosomes transfer their newly synthesized proteins into the lumen of the RER where they undergo structural modifications, such as folding or the acquisition of side chains. These modified proteins will be incorporated into cellular membranes, the membrane of the or those of other organelles are secreted from the cell, such as protein hormones, enzymes. The RER also makes phospholipids for cellular membranes. If the phospholipids or modified proteins are not destined to stay in the RER, they will reach their destinations via transport vesicles that bud from the RER's membrane. Figure 1. Since the RER is engaged in modifying proteins, such as enzymes, for example, that will be secreted from the cell, you would be correct in assuming that the RER is abundant in cells that secrete proteins. This is the case with cells of the liver, for example. The smooth endoplasmic reticulum, SER, is continuous with the RER but has few or no ribosomes on its cytoplasmic surface. Figure 1. Functions of the SER include synthesis of carbohydrates, lipids, and steroid hormones, detoxification of medications and poisons, and storage of calcium ions. In muscle cells, a specialized SER called the sarcoplasmic reticulum is responsible for storage of the calcium ions that are needed to trigger the coordinated contractions of the muscle cells. Career connection. Cardiologist. Heart disease is the leading cause of death in the United States. This is primarily due to our sedentary lifestyle and our high trans fat diets. Heart failure is just one of many disabling heart conditions. Heart failure does not mean that the heart has stopped working. Rather, it means that the heart can't pump with sufficient force to transport oxygenated blood to all the vital organs. Left untreated, heart failure can lead to kidney failure and failure of other organs. The wall of the heart is composed of cardiac muscle tissue. Heart failure occurs when the endoplasmic reticula of cardiac muscle cells do not function properly. As a result, an insufficient number of calcium ions are available to trigger a sufficient contractile force. Cardiologists. Cardi equals heart. Ologist equals one who studies. Are doctors who specialize in treating heart diseases, including heart failure. Cardiologists can make a diagnosis of heart failure via physical examination. Results from an electrocardiogram, ECG, a test that measures the electrical activity of the heart, a chest X-ray to see whether the heart is enlarged, and other tests. If heart failure is diagnosed, the cardiologist will typically prescribe appropriate medications and recommend a reduction in table salt intake and a supervised exercise program. The Golgi apparatus. We have already mentioned that vesicles can bud from the RER and transport their contents elsewhere. But where do the vesicles go? Before reaching their final destination, the lipids or proteins within the transport vesicles still need to be sorted, packaged, and tagged so that they wind up in the right place. Sorting, tagging, packaging, and distribution of lipids and proteins takes place in the Golgi apparatus, also called the Golgi body, a series of flattened membranes. Figure 3. The receiving side of the Golgi apparatus is called the cis face. The opposite side is called the trans face. The transport vesicles that formed from the RER travel to the cis face, fuse with it, and empty their contents into the lumen of the Golgi apparatus. As the proteins and lipids travel through the Golgi, they undergo further modifications that allow them to be sorted. The most frequent modification is the addition of short chains of sugar molecules. These newly modified proteins and lipids are then tagged with phosphate groups or other small molecules so that they can be routed to their proper destinations. Finally, the modified and tagged proteins are packaged into secretory vesicles that bud from the trans face of the Golgi. While some of these vesicles deposit their contents into other parts of the cell where they will be used, other secretory vesicles fuse with the plasma membrane and release their contents outside the cell. In another example of form-following function, cells that engage in a great deal of secretory activity, such as cells of the salivary glands that secrete digestive enzymes or cells of the immune system that secrete antibodies, have an abundance of Golgi. In plant cells, the Golgi apparatus has the additional role of synthesizing polysaccharides, some of which are incorporated into the cell wall and some of which are used in other parts of the cell. Career connection. Geneticist. Many diseases arise from genetic mutations that prevent the synthesis of critical proteins. One such disease is Low disease, also called oculocerebrorenal syndrome, because it affects the eyes, brain, and kidneys. In Low disease, there is a deficiency in an enzyme localized to the Golgi apparatus. Children with Low disease are born with cataracts, typically develop kidney disease after the first year of life, and may have impaired mental abilities. Low disease is a genetic disease caused by a mutation on the X chromosome. The X chromosome is one of the two human sex chromosomes, as these chromosomes determine a person's sex. Females possess two X chromosomes while males possess one X and one Y chromosome. In females, the genes on only one of the two X chromosomes are expressed. Therefore, females who carry the Low disease gene on one of their X chromosomes have a 50-50 chance of having the disease. However, males only have one X chromosome and the genes on this chromosome are always expressed. Therefore, males will always have Low disease if their X chromosome carries the Low disease gene. The location of the mutated gene, as well as the locations of many other mutations that cause genetic diseases, has now been identified. Through prenatal testing, a woman can find out if the fetus she is carrying may be afflicted with one of several genetic diseases. Geneticists analyze the results of prenatal genetic tests and may counsel pregnant women on available options. They may also conduct genetic research that leads to new drugs or foods, or perform DNA analyses that are used in forensic investigations. Lysosomes. In addition to their role as the digestive component and organelle recycling facility of animal cells, Lysosomes are considered to be parts of the endomembrane system. Lysosomes also use their hydrolytic enzymes to destroy pathogens, disease-causing organisms that might enter the cell. A good example of this occurs in a group of white blood cells called macrophages, which are part of your body's immune system. In a process known as phagocytosis or endocytosis, a section of the plasma membrane of the macrophage invaginates, folds in, and engulfs a pathogen. The invaginated section, with the pathogen inside, then pinches itself off from the plasma membrane and becomes a vesicle. The vesicle fuses with a lysosome. The lysosome's hydrolytic enzymes then destroy the pathogen. Figure 4. Section summary. The endomembrane system includes the nuclear envelope, lysosomes, vesicles, the RER and Golgi apparatus, as well as the plasma membrane. These cellular components work together to modify, package, tag, and transport proteins and lipids that form the membranes. The RER modifies proteins and synthesizes phospholipids used in cell membranes. The SER synthesizes carbohydrates, lipids, and steroid hormones, engages in the detoxification of medications and poisons, and stores calcium ions. Sorting, tagging, packaging, and distribution of lipids and proteins take place in the Golgi apparatus. Lysosomes are created by the budding of the membranes of the RER and Golgi. Lysosomes digest macromolecules, recycle worn-out organelles, and destroy pathogens. Thank you for watching BookTube channel. Make sure to smash the like and subscribe buttons to receive notifications about new contents.